Hi, my name is Ria, and my topic is exploring biomolecules, structure, function, and analytical techniques, wherein I will be majorly focusing on carbohydrates. So, what are biomolecules? Biomolecules are organic molecules produced by living organisms and are vital for structure, function, and regulation of the body's cells and tissues. They vary widely in size and complexity and are involved in nearly all life processes from energy production to genetic information transfer. These molecules primarily composed of elements like carbon, hydrogen, oxygen, phosphorus, and sulfur. They are broadly classified into small molecules like hormones and metabolites, and large macromolecules like carbohydrates, proteins, lipids, and nucleic acids. These macromolecules are the building blocks of life, contributing to structural support, energy storage, communication within and between cells, and genetic inheritance. Carbohydrates are macronutrients and are one of the main ways by which our body obtains its energy. They are energy-rich organic compounds and are primarily composed of carbon, oxygen, and hydrogen. These are essential nutrients found in grains, dairy, bread, fruits, and vegetables. Basic structure of carbohydrates. They're organic compounds made of carbon, hydrogen, and oxygen. They can be divided into four major groups depending on the degree of their polymerization, and they link through glycosidic bonds. Now, glycosidic bonds are covalent bonds formed between anomeric carbon of one sugar and a hydroxyl group of another during a hydration reaction. These bonds are essential for forming disaccharides and polysaccharides. The type and arrangement of glycosidic bonds determine the structure, function, and digestibility of carbohydrates. Carbohydrates primarily contain two functional groups, that is hydroxyl and carbonyl group. Hydroxyl groups are present in multiple locations and they make carbohydrates highly soluble in water, while carbonyl group can be an aldehyde or a ketone depending on where they are placed in the carbon chain. Types and forms of carbohydrates. So, carbohydrates can broadly be classified into two main types, that is, simple carbohydrates and complex carbohydrates. Simple carbohydrates are composed of one or two sugar units, while complex have multiple sugar units. Simple carbohydrates are further divided into monosaccharides and disaccharides, while complex carbohydrates are divided into oligosaccharides and polysaccharides. Monosaccharides are uh, Monosaccharides are carbohydrates containing one sugar units. So they're the smallest of the carbohydrates. Because of their small size, they have an important role in metabolism and digestion. They are of three types: glucose, fructose, and galactose. Glucose is used for energy through glycolysis, or it's stored as glycogen in the liver and muscles for further energy needs. Fructose plays a key role in metabolism as an energy source and in the synthesis of glycoproteins and glycolipids. Galactose is crucial in human metabolism as it serves as an energy source and a building block for complex molecules, particularly in early development. It's essential for energy production, storage, and synthesis of various important cellular components. The second type of simple carbohydrates are disaccharides. They contain two sugar units bonded together. Three examples are maltose, sucrose, and lactose. Maltose is made up of glucose bonded with glucose. It's an important disaccharide in cellular metabolism, primarily because it serves as a readily available source of glucose for energy production and other cellular processes. Sucrose is made up of glucose plus fructose. It's a disaccharide and it's essential in cellular metabolism as a major energy source and signaling molecule. It is broken down into glucose and fructose, which enters glycolysis to produce ATP. It also helps in regulating gene expression, plant growth, development, and stress response. Lactose is made of glucose and galactose, and it's composed and it plays an important role in metabolism, especially in infants and young mammals. It serves as key energy source, providing glucose for ATP production through glycolysis. Complex carbohydrates are divided into polysaccharides and oligosaccharides. Polysaccharides are carbohydrates that contain multiple sugar units. They are three examples are starch, glycogen, and cellulose. Starch is made up of long chains of sugar molecules that are connected together. The linear polymer amylose is the most basic form, and amylopectin is a branched form. Our main source of carbohydrates is starchy foods, which play an important role in a healthy diet. Potatoes, bread, rice, pasta, cereals are examples of starchy food. Glycogen. Glycogen is a highly branched polymer of glucose. It serves as the body's primary carbohydrate storage form, predominantly found in liver and muscles. When needed, glycogen is broken down through glyconeolysis to release glucose, which enters glycolysis and cellular respiration to generate ATP. Fiber includes carbohydrates and other structural substances in plants that cannot be digested by human enzymes. It's made by plants to provide protection and structural support. Dietary fiber, though a carbohydrate, is not digested into glucose but plays an important role in metabolism and cellular health. Oligosaccharides are molecules that contain sugar units ranging between 2 to 10. They're not as big as polysaccharides, but they're not as small as monosaccharides or disaccharides. Examples can be raffinose. Carbohydrates have an important role in metabolism and cellular processes. They aid digestion, supporting a healthy gut microbiome. They are primary source of energy for the body and have an important role in cellular respiration. They help maintain homeostasis and support growth, repair, and help in metabolic functions. They form the structural backbone of DNA and RNA through the sugar deoxyribose and ribose. They help in cell recognition and communication, support cell wall and cell membranes, and are building blocks for nucleotides and amino acids. Techniques used to study carbohydrates. So, chromatography, it depends on separation uh, that relies on differential interaction of carbohydrates with stationary and mobile phase. Uh, First example is gas chromatography, which gas chromatography separates volatile components of a mixture based on their boiling points, volatility, and interactions with the stationary phase coated inside the column. Since carbohydrates are typically non-volatile compounds and thermally unstable, they must first be derivatized, that is, chemically modified into more volatile compounds before analysis. They can be used for qualitative and quantitative analysis of monosaccharides, etc. Second is high performance liquid chromatography or HPCL. HPCL separates compounds based on the differential interaction with stationary and mobile phases under high pressure. They're suitable for analyzing carbohydrates without derivatization, especially when using specific columns like amino column, ion exchange columns, or size exclusion columns. The third is thin layer chromatography, which separates compounds based on their affinity for a thin layer of stationary phase coated on a plate, and their solubility in the mobile phase. Simple, it's a very simple and cost-effective method for qualitative analysis, and it allows the separation of sugars based on differences in polarity and hydrogen bonding. Second method is spectroscopy, that is used for analyzing and studying carbohydrates. First example is UV spectroscopy, in which the absorbance of UV or visible light by a substance is measured. Carbohydrates do not naturally absorb strongly in the UV range unless they are chemically modified. Quantification of total carbohydrates or reducing sugars can be done using spectroscopy. Infrared spectroscopy, um, it is the absorption of infrared light, which causes vibration in chemical bonds, is measured here. It's used for monitoring chemical modification in carbohydrates. Then Raman spectroscopy, it's used for studying aqueous carbohydrate solutions and identifying specific sugar conformations. Centrifugation, separating and purifying carbohydrates containing components from complex mixture. It's of two types: density gradient centrifugation and differential centrifugation. And electrophoresis, separation is based on their size and electrical charge using an electrical field. First is capillary electrophoresis. Here, the charged molecules are separated based on their size to charge ratio as they migrate through a thin capillary under high voltage. It's used for monosaccharide and oligosaccharide profiling, and polyacrylamide gel electrophoresis or PAGE is used for proteins. It can reveal glycosylation differences based on altering mobility. And carbohydrate containing macromolecules can also be and can also be studied using PAGE. And that's it. Thank you.